Good afternoon. Thank you for joining us today at the Seminars in Oncology series that we restarted after the summer break. And it's my distinct pleasure to introduce Sarah Hill, who is one of our junior faculty and rising stars in Dana-Farber. And Sarah did almost all her training here at Harvard, starting with undergraduate and PhD, and then also did a residency at the Brigham and PhD with David Livingstone. So she's one of the preparation age kids. She did have a detour to Oxford University to do a master's thesis with Kim Naismith. And she also got a Rhodes Scholarship during her training. And Sarah started her lab only a year or two ago, I think, very recently. And in the middle of the pandemic, which is not easy for anybody. And she's also got several awards already for junior investigator, including the AACR Cancer Research Early Career Award and also the Scholar Retaining Award in Ovarian Cancer Research. And today, Sarah will discuss her work on dissecting high-grade serous ovarian cancer therapeutic response using novel patient-derived organoid cultures. So thank you, Sarah.
Awesome. I'm just going to share my screen. Okay. Sorry, it's coming. Okay, can people hear me? Somebody just give me a thumbs up in the chat.
Yep.
Yep, perfect. Awesome. Cool. Okay. Thank you so much for having me. When I give this talk, what I like to do is just start out with a little introduction to ovarian cancer, because I know that's not what most of us are focusing on. And then I'll talk a little bit about why we've taken the direction that we have and sort of where the lab is going. So for ovarian cancer, it is not the most common of all cancers. In 2021, here, there are estimated to only be 21,000 new cases and 13,000 deaths. But if you look over time, this is from the American Cancer Society, between 2014 and 2018, per 100,000 people in the United States, ovary was the 6th biggest killer. So it's right next to pancreas, which is also somewhat rare, and right next to some of the more common tumor types. And the reason for this high death rate is really twofold. There are very limited treatments for ovarian cancer. There hasn't been anything really new and just super successful for a long time. Right now, it's still surgery and chemotherapy. There are PARP inhibitors out there, but I think the impact of those still has yet to be seen for the general sporadic tumors. And then the other big problem is that this is really a disease of prevention, and we have no means of early detection. So when these patients come in, they will actually come in at stage 3 or stage 4 and be incredibly difficult to treat with a belly full of tumor. And we will do the best we can with chemotherapy and surgery, but even then, there's still a lot of tumor left in the belly, and most patients will recur. So when we talk about ovarian cancer, many people will hear the word ovary and think reproduction, so something like germ cells, but that's not what my lab works on. We work on epithelial tumors, and these are still really categorized as histologic subtypes. So these are the 5 most common ones. We have high-grade serous, which comprises 70% of all ovarian cancers. This is the most common. It's also the biggest killer, and most of these patients will actually succumb to their disease within 5 years of diagnosis. There's also endometrioids. These are not endometrial tumors from the uterus. These are tumors that look like endometrial glands that actually form in the ovary. Very interesting, very different biology. Clear cells, I'm sure you're thinking these look just like clear cells of the kidney. They sort of do. Mucinous, these are very rare. Very few mutations, nothing to treat these. Now we have a lot of models in the lab, so that's an exciting type to work on. And then low-grade serous, so these are P53 wild type as opposed to the P53 mutant high-grade serous. We're extremely excited about the low-grade serous. It's only 5% of patients, but they tend to be very young women, sometimes in their 20s, and there are no treatments. So our lab has a lot of projects on these, and we're trying to do better because they really have nothing. But today, this talk is going to focus on high-grade serous ovarian cancer. And the reason is, when I was doing my PhD with David Livingstone, the TCGA data set started to come out for ovarian. And it indicated that 50% of these tumors have some sort of genomic alteration in a DNA damage repair gene. Now, whether or not that confers a functional defect is sort of something that's still on the table. But I was excited because, as many of us know with David, it's BRCA1 all day, every day, 365 days a year. So this really drew me into the field because, as you can see, many of the genomic alterations are in BRCA1 or BRCA2, which are the big the breast and ovarian cancer genes, or in some of their binding partners. And then there's a myriad of other things. But this was exciting because in David's and other DNA damage labs, a lot of biology has been worked out for BRCA1 and 2, especially in the DNA damage repair pathways. So the two main ones that people focus on, and this is by no means the only functions for these proteins, but the two big ones are homologous recombination, repair of double-stranded DNA breaks, and then protection of stalled replication forks. And so we don't need to go into every detail. David and others have worked out all of the molecular pathways here to start with. There's plenty still left to do, but you'll notice a lot of binding proteins for these large tumor binding proteins like RAD51. And if you go back here, you'll see these binding proteins like RAD51C. So if you harbor a mutation in any of the binding partners, you can also have defects in these pathways. And the exciting thing now is that there are targeted therapies for these pathways. So you've got PARP inhibitors, which could hit both. There's ATR inhibitors, WEE1, et cetera. So the big question in the ovarian field, like one of the dominating things that people are working on, is how do you identify who could possibly respond to some of these DNA damage repair, defective targeting agents, and what do you do for those patients? Is it just PARP inhibitors? Who's going to respond to what? So the field has started to shift towards using patient-derived organoid models in a way that you can actually do functional assays on them to predict patient response. Now, I'll talk a little bit about why I don't think that's the best idea. Right now, a lot of people are very much focused on just using tumor organoids. So what they do is they'll get a tumor out of the operating room. So this is actually an ovary that is full of cancer. This is not what your ovary should look like. This is the histology of the tumor. They'll ground it up and put it in a special media. There's a lot of different medias out there. That's another problem in the field. And eventually, in that media, these little tumor spheres will form that look like this. And this is just a histologic cross-section of one of those. And you can try all kinds of functional assays on these to see if you can match patient response, learn about biology, kind of the sky's the limit on these. So in terms of functional assays, what's out there today, the most obvious one that labs all over the world are trying in different ways is just to directly test the drug on the organoids and see if you can match the patient response. This is very unlikely to work. Many have already published on this. So this is a paper from Hans Klaver's group. He's kind of the father of the organoid field. And they have more patients in this paper. I think there's like 24 in this particular paper. And for these patients in this list and in this sensitivity curve, what they did is these are patients who got neoadjuvant carbotaxol. That's a pretty classic standard therapy. Then they went to surgery, and the surgeon plucked out all of the visible tumor in what's called a cytoreduction. And Klaver's lab actually got some of that tumor and grew organoids. So these are the 7 patients they grew organoids from. And they did sensitivity testing on those organoids to the standard neoadjuvant treatment, which is carbotaxol. These are the dose curves for the carbotaxol. And what they did is they figured out what the area under this curve was, and they tried to match that to different metrics of clinical response in these patients. So pathology score, CA-125, radiology, or survival. This was done with cell titer glow as the read. I'm not sure that's the best method. And they also only have one site from each patient. So they have sort of a mixed bag of response. This really high area under the curve, the patient had like all these different metrics that showed no response, didn't survive well, that's fine, that's a good match. But then these other 6 patients had sort of a mixed response, and it wasn't clear like how well the organoids were predicting. And in the rest of the paper, they go on to show that really they should have tested multiple sites because you needed more sites to understand response. They did a lot of DNA sequencing, and that didn't really perfectly match response either. And so that's sort of what's coming out with these right now. I don't know how well this is going to work. I know we and others are working on different metrics of reading this sort of cell death readout, and we'll see how that goes in terms of matching response in the future. That's one way. The other thing is you can do functional assays to test the ability of tumor cells to perform some sort of task, whether it's looking at stalled replication fork protection in a BRCA defective tumor, or if you've got a KRAS mutant low-grade serous, what kind of functional assay can you do for that? So this is just one example. This is a DNA fiber assay that looks at replication stress. There are many ways to perform this assay. This is just one example. So you can add these different components in different ways to ask different questions about a fork. But in this example, you're going to add 2 nucleoside analogs, so IDU, then CLDU. You'll wash them out, and then you'll add some sort of DNA damage repair agent. It can be an ATR inhibitor, a PARP inhibitor, platinum, some combination thereof. And then the readout when you do it this way is looking at, can the tumor cell protect its stalled fork? If it can, then when you look at the ratio of the green to the red tract of DNA, which you've laid out on a cover slip, the green and the red should be equal. But if the tumor cell cannot protect its forks, then this second nucleoside analog, the tract here, will be degraded, so it'll be shorter. So that's one way to measure this. You can also mess around with the way you add these and how you add the drugs and sort of change this around to ask a different question. But in one of the earlier papers, this is just one of the ways this was used. So these are organoids from patient number 1 and patient number 2. And this is a fiber assay in this patient, and then these are drug sensitivity curves for the organoids from this patient. And what was done here is 3 different biologic replicates of the DNA fiber assay. So repeat 1, 2, 3. Each Christmas tree is a repeat. Each dot on here represents a single fiber that was counted, and it's basically just representing the ratio of the red to the green, so the IDU to the CLDU. And here you can see this red in an example fiber got chewed up, so the red to the green is less than 1. So this would say this person has a fork protection defective tumor. It might be sensitive to some of these DNA damage repair agents. And when you look at the sensitivities of the organoids to platinum, which is standard, a CHEK1 inhibitor or an ATR inhibitor, these are sort of arbitrarily drawn sensitivities, and this was done with cell titer glow. So those are already kind of inherent problems, but with arbitrary sensitivities, this was deemed sensitive to all 3 agents, and that sort of matched with the fiber response in these organoids. Unfortunately, it didn't match the patient. This patient, she came in, she had an unresectable tumor. The surgeon opened and looked and couldn't do anything. So she went to neoadjuvant chemo, and this tumor is actually post-neoadjuvant chemo where the organoids were made. When she went back to surgery, the surgeon again could not operate. So she never got a full resection, and she actually died rapidly. So I don't know how predictive these organoids would have been. This patient is a totally different story. So her organoids are a little bit different. You can see for her fibers, the red and the green look pretty equal. So you'll see that that's a ratio of 1 across all 3 replicates, meaning that's a fork stable tumor. And when you look at the drug sensitivities down here, this looked resistant to platinum, CHEK1 inhibitor, ATR inhibitor, except this patient lived longer than this one. So, I think right now, just using tumor organoids and functional assays, it's just not been well worked out. And in my lab, we just have kind of come to think that ovarian tumor organoids that are made up only of tumor cells from one site have a lot of problems. And I don't think they're going to be predictive of patient response by themselves. So I'll show you why that is, and then I'll show you a way we've come up to get around that to both find better biomarkers, but also find more out about ovarian cancer biology. So first, I kind of alluded to this when I was showing Hans Klever's paper. Whatever tumor you take to make an organoid is all you're going to analyze. So when a patient has a cytoreduction for ovarian cancer, the surgeon will open up the belly and look in, and he'll see, or she will see, many, many different sites, and they'll pluck out each individual site. So these are the jars we would get in pathology, and each of these jars contains a unique tumor site that might be totally different than all the others. And when you count, there are 16 individual tumor sites here. Some are really small, like this one. Some are gigantic and need to be in a bag. So the question then is, how many do you need to analyze to predict response in this case? Then, when you look at these giant bags, when you open them up, this is the tumor that you're going to see. So this is the omentum, and high-grade serous ovarian cancer loves to sit down on fat, especially in the omentum. When you look at this particular omentum, this is a ruler that's 15 centimeters long. This is 30 centimeters of pure tumor. So if you're going to try to make an organoid to predict patient response, I would love to know where you would like to take it out of this 30 centimeters, because I know Klever's and my lab, we've all shown that there are different responses within these multiple sites throughout this huge mass. This is a different omentum, different style of ovarian cancer. This is a more miliary disease, meaning there are little teeny tiny balls of tumor here, 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 here. I could do this all day. So which one of those do you take to try to pick patient response? Again, it's just an inherent problem. So it's multi-site, it's giant masses. What do you do with that? So that's issue number one. The other issue, when you go back to the clinic and really look at these tumors, when these patients die of ovarian cancer, it's almost always due to a bowel obstruction. And we never sample these because nobody's going to go to autopsy. So like whatever we're testing at the end is usually some sort of like superficial met that we can just easily get to through biopsy. But when you look at these, I want to really look in at these tumors. So this is a colon. It's been opened up. It's usually a tubular structure. So you can sort of see the fat on the outside. This is the colonic epithelium on the inside. This ball that I'm pointing to here, and then this one that's been opened, those are bowel mets. They are the ones that caused the bowel obstruction and killed this patient. And when you look at these, this is the histology of the tumor. So you'll see this blue stuff. Those are tumor cells. There's more in there though. Then there's a lot of this pink stuff. That's not tumor. And then this is the colonic epithelium. So when you zoom in on a little area like this over here, this is your zoom in, you can see these are nice nests of tumor cells here and here. But this tumor is not all tumor cells. So you've got this beautiful surrounding of immune cells, stroma, and collagen. And if all you're looking at when you're trying to understand this tumor is the ball of tumor cells, you're never going to understand how this patient's going to respond to therapy. So for my lab, this has sort of driven us in a different direction than just trying to study tumor cells alone. Instead, we're using complex ex vivo co-cultures that contain everything to really ask about the conversation between these tumor cells, immune cells, and stroma. And the two fundamental questions that we're going after with this are, first, how does the conversation between tumor cells, stromal cells, and immune cells change as the tumor cells themselves are being taxed with different types of treatments? So for example, if you have a BRCA mutant high-grade serous and you give it platinum, initially maybe it had the defect that made it platinum sensitive, but that platinum has now driven it to be platinum resistant. Those two types of tumor cells will have a lot of different interactions with the stroma and the immune cells around them. And trying to understand that is going to be really critical in preventing these really broadly resistant environments. And that's our second question. How do we prevent the development of these broadly resistant tumor microenvironments that have so much stroma and are so resistant to these therapies that can't even penetrate? Can you give tumor intrinsic targeting drugs combined with something that will, prevent a problematic tumor normal interaction? Can you do immune therapies plus, a tumor intrinsic drug? Those are the questions we're trying to address. And so, as I said, we're doing this using an ex vivo co-culture system. This is pretty straightforward. The patients will come in. So this is just the CT scan from an average interval debulking. The patient still has omental mets here. There's a giant pelvic mass down here. She'll go to surgery. The specimens will come out. We'll get some sort of tumor, might be multi-site. We'll do mechanical dissociation. Our lab is really heavy into mechanical dissociation now. We definitely don't want to do anything that will damage the immune cells and the stromal cells because we want everything. And then we'll get little single cells or groups of cells and mix them up in, now it's very different medias depending on what the question is. So if we're asking about macrophages, we use a slightly different growth factor setup. If we want to look at activated T cells or non-activated T cells, we'll add different things. And then we'll add drugs into the mix. It could be an immune checkpoint agent, could be a small molecule therapy, could be standard chemo. And we'll ask, how are these different cells interacting with each other? And then the other cool thing is you can actually manipulate the organoids or the stromal cells or what have you, and then add them back and ask how some sort of genetic perturbation might also change that interaction. So that's what we're doing in the lab. This is what the co-cultures look like. So on the left, this is the tumor that I was showing you. And then on the right, you've got a co-culture. So these big purple cells, those are your tumor cells. They're represented by these little spheres here and here and here. And then these single immune cells, you can see these smaller, tiny single cells. Those are all the immune cells permeating. And then this picture, I haven't gotten a later picture. This is a picture right after plating, but within about 2 hours, these stromal cells sit down and form a really nice bed for these tumor and immune cells to sit down on. And we can start to ask what happens between, each cell type as time goes by, who's interacting with who, what does, different stress do to each of those interactions, et cetera. So what I'd like to do is just take you through some of our preliminary work to show you that this system does work, and then just give you a sense of where the lab is going. So the first thing I always get asked is, do these, crushed up co-cultures actually match the parent tumors? So we've gone about this in multiple different ways. The first one is always flow cytometry. We prefer flow. So this is just a paired set of 10 tumors and their co-cultures. So on the left in each of the pairs is a parent tumor. On the right is an organoid co-culture. Each of the bars in these represents a different immune cell type, and it's given as a percent of CD45 positive cells. You can also do absolute immune cell numbers. Sometimes they can be a little bit low if it's like immune cold tumor. So I just show it this way so people can see the bars a little bit better. And I'll draw your attention to just a couple different things here. If you look at the red and the yellow, those are the CD4s and the CD8s. So in the organoids, we always get expansion of the CD4s and 8s, even if we don't put any growth factors in. So if we're doing something that's very immune focused, we do try to be cognizant of that and not leave these in culture too long, just in case we're expanding something that's non-specific, but it's just unavoidable. The second thing is, this was our early work. So we had something kind of unfortunate happen. If you look at the green, the blue, the orange, and the gray, those are gonna be myeloid cells, like macrophages or dendritic cells. And initially those were dying off relatively quickly. Now we've been able to tweak the media a little bit and get them to survive longer because they are really bad actors in the tumor microenvironment. But you'll see in this paper that was sort of something that was sort of unavoidable. And then the last thing I want to draw your attention to is the purple. So those are NK cells. This is a very NK-centric story, and now my lab's really excited about them, and we've been able to collaborate with Rizwan Romi to sort of take this story forward, and I'll hint at that at the end. But we were surprised. If you look at the parent tumors and the organoids, there are actually a lot of NK cells in each of these, and NKs aren't really targeted effectively by any immune therapy on the market. There's monalizumab that's out now, but that's it. And we'll show you that that's one of the unique things that we found is a way to draw those NK cells into the anti-tumor immune response. So if this wasn't good enough, we also did little ball and stick graphs. So for each of these ball and stick graphs, it's basically the percent of CD45 positive cells that these different cell types comprise in the tumor. So the blue is the tumor, the triangle is the organoid. So for the CD4s, 8s, Bs, and NKs, you get a pretty stable or at least an increase in the cell types. And then, as I said, the myeloids all went down in this first paper, and now we've worked really hard to get them to stay. So we've got some cool small molecules that can play with those. And then the last way we analyzed this was single cell RNA-seq. So this was comparing a parent tumor to an organoid co-culture up here, and you've got a UMAP showing all the different types of immune cells that were taken into the organoid co-culture compared to the parent tumor. And when you overlay them, the organoid is orange, the parent tumor is blue. So you can see all the immune cells are really well represented except for the macrophages. And I'm so bummed I can't show you. We just got another one back today where like the macrophages are all there. So we've definitely improved on this, but even with just that, we were still able to ask a lot of good questions. And the reason I say that is if you look at the T cells, we were really nicely able to divide up all the different T cell subtypes and ask good questions about transitions between each different subtype. And you can see the overlay of the orange organoid to the parent tumor here shows that they're all well represented. So I think the co-cultures are a reasonable match to the parent tumors. And our next question was really, can we detect any kind of bulk response? So we didn't initially go after the stroma, we went after the immune component of this first. So we started out trying to detect a bulk immune response. And to do that, we decided to try to test immune therapies that have failed in ovarian cancer compared to something new that might be slightly better. So we used pembrolizumab, which is anti-PD-1. We did an anti-PD-L1, and those have not done well. These are not effective alone in ovarian. And then we used a novel bispecific anti-PD-1, PD-L1 that we got from Eli Lilly. And the idea was that this isn't going to be a game changer, but our hope was that it's going to at least be incrementally better. And if we can find the mechanism behind how this thing might be slightly better than these two single targeting agents, we can look for small molecules that can better hit that mechanism and better draw the immune response back into these tumors. So what we did is we generated 13 co-cultures, treated them with either isotype control or one of these 3 different immune checkpoint antibodies, and then we used interferon gamma ELISAs just to measure a bulk immune response. So this is just like a 96-well plate at the bench, looking at the supernatants from the co-cultures and asking how much interferon is in them. We actually still do this. It's an incredibly effective screening method for any kind of drug that can be tested. And we had 13 tumors. So in each of these bars, The little black dots represent a different tumor. And what we found is the level of response was different, but we always saw the same trend. And to try to put all of this in a single bar graph, we actually just had to normalize to the IgG control. So the y-axis is the average interferon gamma fold change by ELISA over the IgG control. And what you'll see going from IgG to the single targets to the bispecific always induced the strongest immune response. So we were happy about that because that's sort of what we thought it would be. It wasn't huge, but it's going to be enough. I'll show you to draw out a mechanism. And then one of the questions that we got was, is this bispecific just an additive effect of adding together pembro and PD-L1? So we did that in some of the cultures, which you can see here, and it's not. This bispecific definitely has unique properties. And there's a paired paper that came out right around the same time as ours, where they show this bispecific does exactly what you think it would. It spatially draws together the tumor and immune cells. And then it can also simultaneously engage multiple receptors on the same or different immune cells. So that's what's giving it these kind of superpowers, so to speak. And then if you do any of these comparisons for t-tests, all of these differences are going to be significant. So everything that you want to see is definitely different. This is the time then, this is a bulk immune response in a co-culture. The question then is this matching the bulk immune response or sort of the bulk immune state of the parent tumor? So to do that, we did bulk RNA sequencing on all of the parent tumors from which we generated these organoid co-cultures, and we compared it to the ELISA. So the ELISAs are here. This is a heat map. The more red it is, the stronger the interferon that we saw. And what you'll see is here's an interferon gamma signature score for each individual tumor. This was generated by looking at a set of 12 or 13 genes associated with interferon gamma expression. And so the higher the score, the higher the interferon signaling in that parent tumor. So if you look at the lowest responding tumors here and here, they have the lowest interferon gamma signature scores. And then if you look at our highest tumor here, that has the highest score. And then the rest that are like higher are kind of in between. So this is like a blunt instrument, but at least it's not a horrible match. And we felt pretty good about that. Any discrepancies I think are definitely going to be sampling error. Just we took like 1 centimeter to make the co-culture and another centimeter to make the bulk RNA sequencing. So I think that would be how you would see differences. But overall, I think, we were really happy with the results to know that we could detect a bulk immune response this way. So our next question was, what cells are causing this interferon to be secreted? Who's doing this? Who's activated by the immune therapies? So to do this, you could ask it a lot of ways. We did it with flow cytometry. So this has been an amazing collaboration with Susan Lazo and the flow core. She's helped my lab set up now like 5 or 6 14 color flow panels that are really our workhorse panels. We use them every day, like literally yesterday, we were sitting there for hours doing this. And what we do, we'll stain the co-cultures and we'll gate initially just on viable CD45 positive cells. So x-axis is CD45, y-axis is one of the zombie dyes. So if you have zombie dye, you're dead. And so we have a good amount of viable CD45 positive cells. And what you'll see in here also, there's a lot of dead tumor cells. It's not worth it to try to sort them out. When you get a tumor sample, there's always tumor that's dead. It's necrotic. It didn't have enough oxygen. Every minute it equals a macrophage. So when we get these tumors, we just go for it. And there's just going to be some dead tumor cells in there. And we've just gone forward and haven't had issues with flow. But that's what that is, if there's any curiosity about it. Then we'll gate either for a whole myeloid panel, which I'm not going to get into because we have kind of limited time, or we'll gate T cells versus NK cells. And then for the T cells, we'll draw out the CD8s and the CD4s. So like, we're not just gating on 4 cells, and you can definitely get good separation. And then from this point going forward, you can ask any question you want. So we have like an entire NK cell panel asking about like anergy or cytotoxicity. For the T cells, you can ask about activation, exhaustion, et cetera, et cetera. So that's what we did here. And I'm just going to give you the punchline. The bispecific antibody was consistently activating the T and the NK cells, and it was drawing the NKs and the CD8s into a cytotoxic state. So I'll show you. With the flow, we did the exact same thing we did with the ELISA. So the y-axis is always the fold change compared to the isotype control because the changes were different levels. And so you'll just see that this is just kind of like a weird sort of small change. That's the reason, it's fold change. The levels were too different to put on a single bar graph. And so what you see here, these are Ki67 interferon gamma double positive cells. So for the CD4s, the CD8s, and the NKs, the bispecific always induces the strongest activation in each type. And that was exciting because, again, nothing ever induces NKs to be drawn into the fight. So we were excited about that. And then in looking at CD107A, this is a marker of cytotoxicity. Only the CD8s and the NKs showed a response. It's small because it's the fold change, but you can see the bispecific drawing these cells into a cytotoxic state in both of these cases. So that was exciting. We knew that we could figure out, okay, these are the cells that are responding. We can see the response, but it's not good enough. So we wanted to go after the mechanism now because that's the most exciting thing. And that's the way we could find new drugs. So to do that, we've jumped into single cell RNA sequencing. This is the setup that now we're kind of traditionally using. So we'll take our organoid co-cultures, and in this case, we treated with isotype control, Pembro, PD-L1, or the bispecific. You can imagine doing this in a different way. So like you can do small molecules, you can do small molecule immune therapy combos, whatever you want to do. We do the standard treatment time, and then we'll go to Susan in the flow core and get help sorting the viable CD45 cells out. We'll hash so that we can multiplex. You can also hash before it's kind of however you want to do it. Pool everything, do the 10x reaction, and then analyze. So this analysis is like, there's still stuff to be pulled out of it just from like one tumor. What I'm gonna do today is just show you the two key findings in the NK and the CD8s, but you'll see there's so much you could do with it. So before we get into that, I just want to show you the controls. These are all of the cells that we sequenced. So this is just the map of every kind of immune cell we had. And the most important thing is, when you draw this map for the isotype control or the 3 immune checkpoint blockade agents, it looks exactly like this. So we didn't drop any cell types when we did each individual treatment, which was important because you know then at least your comparisons across treatment match. And then the other exciting thing here is when you look at the different types of T cells, you can see you've got this beautiful representation of like conventional CD4s versus regulatory T cells, all the different types of CD8s. You can actually then draw all of these into different clusters. So we have like 5 different CD8 clusters, and I'm saying this because obviously it's going to be important. And you can do all kinds of different analysis within each type of CD4 or CD8. So in this way, I'll show you, we were essentially able to figure out which small subset of CD8 T cells was responding to the immune therapy in this case. So let's start with the NKs. For the NK cells, it was a really pure population in this analysis, and now we're just like incredibly excited about NK cells because of this work. Our analyst was Cheng-Shin Wan. He's a student, or was a student with Shirley Liu and Miles Brown. And what he did is he created an NK cell activation score using 22 different genes associated with NK cell activation. And he looked at the expression of those genes in all the NKs in each of the 4 treatment groups here. And you can see that the highest score was for the bispecific antibody, and that was the only one that was significant compared to the isotype control. And if you draw out all 22 genes here, this is like not rocket science, it's a very night and day difference. Blue means downregulated, red means upregulated. So like clearly if you just look at the bispecific antibody, it's like all red, which is totally awesome. So this is the only therapy that's drawing the NK cells into the fight. And you can sort of segregate them according to function. So it's cytotoxicity genes here, activation genes here, metabolism, et cetera, et cetera. So neither of these immune checkpoint antibodies are doing a lot. So this bispecific had some cool new function, and I'll get to that in just a second. We tried to do this for the CD8s, and we ran into a little bit of a problem. There was a bit of a signal to noise ratio with the CD8s because, as you'll recall, I showed you in the CD8 population, there are all these different clusters, and it turns out only 1 or 2 are actually responding to the therapy and the rest aren't. So when you try to figure out, you know, okay, is there an activation score? You can't do it on the bulk CD8s. So what we did instead is we focused in on 3 of the CD8 clusters. Those were the progenitor exhausted, the naive, and then the terminally exhausted. And this is just a little map of the markers that Cheng-Shin used to define each of these clusters. Now, when you look at these 3 clusters and you ask what number of, or what percentage of cells are they comprising in each of the different treatments, you'll see in the naive CD8 T cells going from IgG to bispecific, the percent goes down. when you look at the progenitor exhausted, going from IgG to bispecific, the percent goes up. And then when you look at the terminally exhausted, it goes down. So it sort of looks like these two might be transitioning into the progenitor exhausteds. And that is actually the case. So Changsha did something cool. He did diffusion analysis over pseudotime. So this is pseudotime on the x-axis going this way. And activation is also going this way. So we looked at activation and exhaustion over time. Exhaustion is increasing on the y-axis. And these are our three clusters. So the orange are the naives, the red are the progenitor exhausted, the blue are the terminally exhausted. So over pseudotime, you can actually see the naive CD8s increasing activation and giving rise to the progenitor exhausted. And you can see the terminally exhausted being drawn into an active state again towards the progenitor exhausted. So that's exactly what we're seeing here. And the bispecific was doing that most effectively. So that was our other major finding. These were the two major responders. And then just to draw that out a little more, because this is sort of hard to swallow if you don't do this kind of math all the time. Here's just one of each of the markers we used. So we used granzyme B to mark cytotoxicity. So you'll see over pseudotime, the naive cells are becoming more cytotoxic. They're increasing in granzyme expression and giving rise to the progenitor exhausted. TCF7 is a marker of naivety. So you'll see lots of TCF7 expression in the naive CD8 T cells, and then it decreases as you go towards the progenitor exhausteds. And then Havoc2 is actually TIM3. So TIM3 is an exhaustion marker, and you'll see there's not much in the naives, but once you get into progenitor exhausted and exhausted, it's really there. So that's how we did the mapping. That's how we came to this conclusion. And then we were able to actually go back and for the CD8s make graphs like this. And what Changsha did is he actually generated cytotoxicity scores and exhaustion scores using only the naive and the progenitor exhausted CD8s, and then it totally made sense. So we looked at the top 50 genes associated with granzyme B expression or interferon gamma expression and used those to make the cytotoxicity score. So if you look at the scores, the bispecific has the highest score compared to all treatments, and it's the only statistically significant, well, it's not the only one, but it's the most statistically significant compared to IgG for granzyme B. And then the same thing for interferon. So at least you have reasonably good cytotoxicity being induced. And then you have for Havoc2, which is TIM3, or PDCD1, which is PD1, you have decreasing exhaustion scores. So it's the same thing, top 50 genes associated with each for expression, and then create a score. So the score goes down going towards the bispecific in each case. So this antibody was causing increased activation, decreased exhaustion, both in the NKs and the CD8 T cells. And our next question was, how? What is the mechanism? So this was like all happening as we were going towards the shutdown, but we got very lucky. I went back and just really combed through all of the differential expression analyses, like all of the comparisons, looking for what's the most exciting thing, where are there small molecules that we can buy that will target these things. And my eye was caught by a protein called BRD1. This is a bromodomain containing protein. Very little is known about this particular one. It has been very understudied, which is great for a young investigator because now we can work on it since there's not a lot out there, which is what we're doing. But I was excited because when I looked at the NK cells in the single cell data, this particular gene, BRD1, was heavily downregulated. It was obvious. This was so far and away the one that was gone. And this is the BRD1 expression level across treatment in the NK cells for the single cell. So you can see BRD1 is really down, and it's statistically significant. I thought that was cool, but I wanted to make sure it could repeat. So I actually generated a completely brand new co-culture from another patient, treated it with all of our different drugs, then ran to Susan like as we were shutting down, and we sorted viable NK cells and sent those for bulk RNA-seq. And lo and behold, this is BRD1 expression in that set of NK cells. The BRD1 was down with the bispecific again. So this was heavily reproducible, and it's going to be an NK story, which is what you're gathering, but I will show you the CD8s. So we took the naive and the progenitor exhausted CD8s, and we looked at BRD1 expression across treatment, and this is just expression level here on the y-axis, and you'll see BRD1 is indeed down with the bispecific, and it's the only statistically significant drop. So we ended up focusing on BRD1. Amazingly, there is a sort of specific BRD1 inhibitor out there called BA299. So we were able to get this BRD1 inhibitor into the lab. We started treating it in co-cultures, and we've done both ELISA data and flow cytometry. So for the ELISAs, this is just a single co-culture treated with either vehicle or the BRD1 inhibitor in combination with our 4 antibodies going across. So blue is always vehicle plus something, orange is always BA299 plus something. And you'll see that the standard thing that happens is the BA299 always increases interferon, which is kind of exciting, except for with the bispecific, because it had already dropped the BRD1 function. So this is a whole new area of investigation for us, because you can imagine the combination possibilities here, but it was exciting because this was a great mechanism that we found for the bispecific. And the way that it's increasing this immune activity, if you look at the flow for the CD8s, the NKs, and I'm not showing the CD4s here, look at PD1, TIM3, or PD1, TIM3 double positive cells, the BA299 is dropping exhaustion markers. So it's dropping them in the CD8s, it's dropping them in the NKs, and it does it in the CD4s as well. So this thing is essentially increasing immune activation, decreasing exhaustion. That's exactly what we saw in the single cell. And we thought that was great, but still not good enough. So we dug in even further to the mechanism. This was in collaboration with Miles and Henry Long in the CFCE. They helped us. We got an NK cell line called KHYG1, and we treated it with either DMSO vehicle or BA299, and we did ATAC sequencing paired with bulk RNA-seq on these treated cells. So the ATAC I always find to be a little nebulous, so I'll take you through it slowly. For the vehicle, what you're seeing here are open up chromatin peaks or closed down peaks. So this is what it looks like in the vehicle. With BA299, you see that the up peaks increase, the down peaks decrease. This means the chromatin is opening up, okay? And Chang Shin was actually able to map different transcription factors to the up and the down peaks. So you can see here, these are the transcription factors that map to the up peaks. These are the ones that map to the down peaks. So you'll see that there are actually some major controllers of immune cell maturity in there, including GATA3, TBX21, and RUNX3. So we found that pretty exciting. And it looks like this BRD1 inhibitor is causing global chromatin remodeling in the NK cells to draw them into this active state. And just a couple examples of that. With ATAC, the readout you'll see is chromatin peaks going through the genome. So this is a gene called EMB, and here's the vehicle treated cells. These are the BA299 treated cells. And you'll see this peak here. It's up in the vehicle, it's down in the BA299, it's closed down, so that chromatin is shut down. And then the bulk RNA-seq, the EMB expression was down. Well, this is exciting because EMB is a marker of naive NK cells. So that means that BA299 is shutting down naivety and driving the NK cells towards mature cytotoxicity. That's what you would want. And then here, this is CDK9. So what you'll see here, this is the CDK9 promoter. On the top, this is vehicle treated. On the bottom is BA299. This peak in the promoter is actually decreased in the BA299. So CDK9 expression was actually being driven down by the BA299, both in the chromatin and we saw it in the bulk RNA-seq. CDK9 is now interesting because now there are papers out showing that CDK9 inhibitors will actually increase the anti-tumor immune response. So it looked like BA299 was going to be an immune therapy player. And we wanted to make sure that these chromatin changes that we were seeing coincided with actual immune changes in vitro. So we took our KHYG1 cells and we did some ELISA and flow-based assays, which is what I'll show you here. So on the left are the ELISAs, and when you just treat KHYG1s with vehicle DMSO or BA299 and do an ELISA on the soup, you'll see the big jump in interferon, and that's great. That fits with what we were seeing in the chromatin. But when you combine those KHYG1s with an ovarian cancer cell line, OVCAR8, this jump is even higher. So these are the OV8s alone with vehicle or BA299, KHYG1s alone, and then here's the combination. So DMSO versus BA299. And you're probably thinking, wait, she just showed us these KHYG1s alone. Look at the y-axis. This is 10,000 picograms per mil compared to 200. So there's definitely a lot going on in these co-cultures, and we'll show you that it's actually increased cytotoxicity in a second. So by flow, what you'll see then is if you do the single NK cells or the NK ovarian tumor cell co-cultures, there's increased CD107A on the KHYG1 NK cells when you add BA299. So here it is increased when the cells are by themselves with BA299, and then here it is increased in the co-culture setup. And then you can also just look at like general markers of activation. So increased interferon alone or increased combined interferon Ki67. So it's the same setup, KHYs alone or the co-cultures on both sides. So the BA299 is clearly causing like a major increase in immune activity in these NK cells. And just to prove that it is increased cytotoxicity, we did set up co-cultures. These are challenging experiments. I think I saw a collaborator on the line who's trying to do this with me right now. Making the NK cell, tumor cell, or NK organoid co-cultures, and then trying to show cytotoxicity is always challenging. The way we do it is we actually read for apoptotic dead cells. You have to get the right cell mixtures, the right time, the right readout. All of this is really challenging, but we were lucky and we were able to do it. So when you grow OV8s together with KHYG1s, either in the presence of vehicle or BA299, and then a few hours later try to measure for apoptotic dead tumor cells, with the BA299, it's always higher. So clearly the NK cells were causing tumor death. It just wasn't easy to get this perfect. So I know it's not like the cleanest data, but it is statistically significant. So we were happy about that. And this is all great. It's in vitro though, and that's the problem. So to get this into a patient, we knew we needed in vivo data. Unfortunately, this happened literally like during the shutdown, and it wasn't the easiest time to do an animal experiment, but we did pull it off. So I apologize for the low tech nature of this. We were able to get a syngeneic mouse model called the Stowe's model. This is made from ovarian surface epithelium, forms gorgeous solid tumors as well as ascites. So it's a perfect anatomic mimic of high-grade serous ovarian cancer. You can do easy IP injections, and it's in an FEVN background, so just very easy to work with. We definitely have continued doing that. So we started out just injecting our mice on day 0. We couldn't do any imaging, so we just monitored them by weight. 18 days later, we started daily treatments of BA299, and we did 18 full days of treatment, and that's when we reached our humane endpoint. Now, interestingly enough, at day 36, we had this like very sad realization that all of our mice were the same weight. So when we went in to like open up the mice, we thought that nothing had happened, but that was not the case at all. So these are the vehicle treated mice. And I just did a shot right of the abdomen as we were opening, because this is what high-grade serous cancer looks like in patients. We had amazing peritoneal deposits, diaphragmatic deposits, bowel mesentery mets, and then here's your bowel obstruction. This is the vehicle treated animal. So that's great. The vehicle treated animals form tumors. It looks exactly like high-grade serous. The BA299 animals had nothing. We were shocked. I was just ready for it not to work given the weights. And what I noticed is actually these animals got fat. And we are not totally sure metabolically what this drug is doing, but the animals just got fat. So, you know, it was a way to sort of like cure your cancer, but give you type 2 diabetes. And I guess that's probably better. So we're working that up now. I think we have a little bit more of an understanding of what's happening, but it was very exciting. And then I told you this was low tech. So here's our measures of the tumor amounts. We actually ended up just dissecting all of the tumor out of each of the 10 animals in the vehicle treated group and the BA299 treated group, just putting it in dishes and trying to visually quantify it, because this is what we got away with. So the visual score is like a 3 for a lot, a 2 for kind of a middle, 1 for limited, and 0 for nothing. So if you look at the tumor volume scores for the vehicle compared to BA299, obviously they go down. And then the ascites volumes we just did by like aspirating the ascites out and measuring it for each of our 10 animals. So you can see that the ascites volume in vehicle compared to BA299 is less. We had a couple that did form nice ascites. And then when you put that all together in one graph, you can see the ascites volume on the y-axis and then the tumor volume score on top of that. So all the vehicle treated animals had high ascites volume, high solid tumor. For the BA299, it was mostly low volume, low solid tumor. And then the last cool finding is that we were able to do flow on both the solid tumors and the ascites and match what we found in vitro. So I'll just show you the solid tumors. For the solid tumors, we ended up grouping the 2 vehicle groups, or the vehicle treated animals into 2 groups, and the BA299 treated animals into 2 groups, just because there wasn't that much tumor and we needed a fair amount for all of our different flow analyses. And so here I'm just showing the T and the NK results. And you'll see the red and the green, just as in the bar graphs I showed you before, are the CD8s and the CD4s. So you can see they expand. The BA299 compared to the vehicle, this drug is causing an expansion in T cells. And then also, if you look at the pink and the purple, that's 2 different NK cell markers. We used 2 because we knew the NKs were so important in this story. They overlap a little bit, but they're not perfect overlaps. So I'm actually showing both of them here. And when you compare the vehicle to the BA299 treated group, you see a nice expansion in the NKp46 purple NK cells. And then if you actually look at PD1, the exhaustion marker on the 2 different NK cell types, you see it goes down with BA299 in both groups here. And then with the CD8 T cells, you also see a nice decrease in PD1. So that's exactly what we saw in vitro. So we were excited. This is a new small molecule immune therapy for high-grade serous. And now we're working more on the mechanism. So that's our future directions. Right now in the lab, we're looking at BRD1 mechanism of action in tumor, immune, and stromal cells. And interestingly, it's definitely got differences. And then we're studying some pretty interesting combinations, either of BRD1 inhibition with immune therapies or standard therapies. And I think that'll show some promise as well. And then the other big focus is just really using this system to try to look at the interaction between either tumor and immune cells, or tumor stroma, or tumor fat. So that's another project. And then I couldn't really help myself. So we've also expanded everything into uterine cancer as well. So there's a lot going on, but hopefully I've convinced you that the co-cultures are a really relevant model, and hopefully something that will draw a lot more good combinations out for these patients who haven't traditionally responded to immune therapies. So quickly in the acknowledgements, I know we only have like 5 or 6 minutes. So thanks to Miles, Shirley, Changshin, Henry, and the CFCE, a lot of the analysis was not possible without them. And then my longtime technician, Katie Lynch, our Lilly collaborator, the flow core, all of our clinical teams, and all of my funders. And then if you're not brave enough to ask a question now, this is my contact information. So we're up on Smith 8. We love to have visitors if you're on site. This is our lab website. And then if you like what you saw, we've actually got a huge organoid and tissue bank. So we're always happy to collaborate. So I'll open it up for questions. I see some.
Okay, thanks, Sarah. That was great. That was really tour de force, and I was looking at the paper meanwhile as well. So we have a number of questions. First question was kind of expected, is like, co-cultures are great and to capture some of the tumor microenvironment, but usually they don't maintain the long-term, the multiple cell types. So how do you maintain them, and how you ensure that the culture medium that is good for one cell type is also good for the other one?
Yes, so I actually have an early independence award from the NIH, and that's the exact focus of my award. So we do both things. We do short-term co-cultures, sort of asking very specific questions about a specific drug, and then you've hit it on the head. Now we've actually got all these kits where we're separating out each type of cell, so macrophages, fibroblasts, NK cells, tumor cells, trying to have separate cultures, and then the idea is to put them all back together. And the hardiest guys are going to be your T and NKs. So the major focus of every media we have now is trying to keep the macrophages going. Like that could be a full-time job. They're so delicate. So that's what we're working on now. And as I kind of hinted, we've played around with the media a lot to keep the short-term guys going, but in the long term, it's hard. And the other thing with the T cells too, you have to be careful because you don't know what it is that you're expanding. And if they're non-specific, then it's pointless.
Yeah, and especially T cells are very sensitive to being activated in culture. Yep. If you have anything that's, they could see, then they get activated. All right. So the other question is also actually related to, have you tried to separate the epithelial and immune cells from the co-cultures and do a fork stability assays to see if the immune cells regulate the fork stability minus cost treatment? And if NK cells also change replication, fork dynamics, and treatment outcome. So that's a long question.
We're doing that now. We're actually doing it now. That's like one of the most exciting things we're doing in the lab. So Rizwan Romi's group, he's very NK cell focused, has been helping us with this. both with primary NK cells, NK cell lines, and then NK cells coming out of the tumor microenvironment. And it's actually a different question in each style of NK cells. So you really have to be careful about where you're getting your NKs, because the ones that come out of the tumor microenvironment have very different issues than something that might just come out of your blood. So it's a great question. It's something hopefully I'll be able to show you in the future.
Yeah, you could try to flush well, hard to flush the patient sample, but anyway.
Yeah.
Okay, so the other question is, let's stay on the co-culture. So have you compared paired versus unpaired co-cultures? I'm not sure what they refer to, paired or unpaired. AB, I don't know if they could clarify, but I don't know. Do you know, or can you answer, or we need?
I'm not totally sure what they're asking.
I don't know what it means either. So
yes.
I don't know what paired and unpaired. Oh, you know what I could guess is that you pairing the patient and the immune cells from the same patient, and maybe not. I don't know if that's what they mean, but I don't think you would want to do that, because then.
Oh, I think this might be what you're asking, and if it's not, please email me. They might be asking about like immune cells not from the same patient. So
yeah,
is this
that's what I mean. That's what I was guessing, that maybe you have a mismatch. So
it's a CAR question. Okay, so yes, we're working with CAR NKs and CAR Ts, and that's actually one of the other really cool and fun things to do, because you can spike those cells into the co-cultures. So the CARs are like totally fine out in vitro. When they hit the macrophages and everything else, it's bad. So that's one of the coolest things we can do, because we can figure out how can we help those things survive, how can we help them penetrate these heavily stromal tumors. So that's one of the, I think, coolest things we're doing right now. So yes, you can. That's yes is the answer.
Yes. So a question, in the mouse experiment, did you confirm that the emerging tumors are actually high-grade serous in nature? And have you done any molecular profiling on them to confirm they have the mutations and the changes you expect to see?
So the STOWS model comes from Barbara Vanderheiden in Canada. She's wonderful, and she's done all of that profiling. I think she has a beautiful Nature Communications paper out. So we actually have isogenic pairs of the STOWS. The original STOWS is P53 wild type, BRCA wild type. So it forms a poorly differentiated, ugly carcinoma secreting lots of mucus. So it's not totally clear if it's more along the clear cell path or not. It looks like a high-grade serous under the scope. Now we've got sets that are P53 mutant, either point mutation or deletion, and we're adding in BRCA mutations. So what we used there was the pure STOWS, because there was no choice. So.
Okay. And related to mice, have you re-challenged any mice from your in vivo studies?
So does this mean, did we keep any alive, or?
I would think so, that, you would treat and then they develop an immune response, and then you would re-challenge them.
No, we're doing more experiments now with like immune small molecule combinations, but those mice from this particular study, we actually sacked them all that day, because they had developed so much ascites that they weren't really able to walk in the vehicle treated group. So that was the.
Okay. And a question related also to the mice. Is there an increase in recruitment of NK T cells to the tumor or peritoneum, or just the activation? And is there any systemic effects of your therapy?
Nothing systemic. So we actually, before we were able to do these experiments, did the toxicity studies here. Nothing was noticed. We didn't notice any toxicity at all, other than the mice got really smart. It's oral gavage. So they always figure it out, and you know which one's going to give you trouble, and they're trouble every single day. But no, there was no obvious side effect other than a little bit of the extra abdominal fat. That was it. And we didn't do any histology. We did just all flow-based assays. So that's it's a, I guess, low-hanging fruit.
Yeah. So the last question here is kind of related to that. Is like, how would you handle a potential side effect of fattening of the mice, which it would be more of an issue in patients, of course.
Yeah, I mean, I think, and especially in high-grade serous, because most of the high-grade serous patients will be, 60 average age. And so I think you will just have, you monitor CA-125, you're probably going to monitor blood sugars, you can monitor, anything marking diabetes. We just don't know. This drug hasn't been used in patients. It's something that we're discussing now. So we'll see if it makes it.
Okay. I think that's all. Lots of questions and very good attendance. So thank you so much. Very nice work.
Thank you.
And thanks for again presenting. So thank you.
Yes, and thank you for having me.
Thanks for everyone for listening and joining us. Thank you. Bye-bye.
Bye.